I'd like to introduce in the next item of our presentation, Dr. Leon Peto, who's a senior clinical research fellow at Nuffield Department of Population Health. And Leon's going to be talking to us about treatments for COVID-19, but in particular the recovery trial. So, Leon, over to you. Hopefully you can speak and hear and see us.
Yeah, thanks. I'll just try and share my slides. Is this that you mentioned that there was a problem earlier?
There was, but give it a go. I can see them. So go ahead.
Can it? Okay, so yeah, thanks for inviting me to speak. Let me just right. So can people still see?
Yep, still see it.
Leon, I'm sharing them for you, so that's fine.
Is that so? Have I got control of the slides? Can you see it?
Yeah.
Leon, I think you've got control of the slides. So just I think you've got control of the slides.
Okay. Yeah. Thanks for inviting me to speak. I'm Leon Peto. As you said, I'm a research fellow at the University of Oxford, and I've been working on the recovery trial since late 2020. But I'm also a microbiologist working in NHS in Oxford occasionally. And I'm going to talk about treatments for COVID-19. I think you've been discussing testing already, and you're going to have something on vaccination later. So I'm just going to talk a bit about treatment, and in particular treatment of people in hospital, because those are the population that recovery was recruiting. I'm only going to mention outpatient treatment briefly, although obviously that's an area where there's been a lot of success as well.
So sorry, Leon, could you switch your camera on? It's gone off.
Let me see. Okay, can you see me?
Yeah.
Okay, and more importantly, can you see the slides?
Yeah, I think I have control of them, so just let me know.
Okay, thanks. So I'm going to talk briefly a bit about the context in early 2020, although if you've had COVID all morning, then you probably don't need much reminding about that. Then I'm going to talk about the recovery trial, and that's partly just for the sort of parochial reason that I've worked on it, and it's the trial I know best, and those particular results, but also because I think it's one of the sort of real UK successes in terms of COVID research. It's had, I think, an outstanding impact in producing sort of clear results for reasons that I'll go into. Also, as a platform trial, the design means that there are treatments compared in the trial, but new treatments were added and treatments were removed when they were found either to work or not, which means that it's now, I think, on about treatment number 15 or 16, but it provides, with the results for recovery, quite a good overview of the major developments in terms of what we've understood about the treatment of COVID. And then also I'm going to mention at the end just a little bit about lessons from recovery in COVID-19 trials in general, because we've, in some ways, with research on COVID, we've leapfrogged our understanding of lots of other common conditions, and we now know much more about treating COVID than lots of other things. And I think some of the lessons as to what worked in terms of generating good evidence for COVID actually would be very beneficial for all sorts of other areas of medicine. So this is just a graph of daily COVID deaths in early 2020. You'll see this just shows deaths in China and the UK, and you can see the sort of early outbreak in Wuhan, which was controlled with fairly drastic public health measures, limiting the number of deaths. And then just the speed of the increase in the UK, where we went in March and in the space of about a month from essentially no COVID deaths to 1,400 a day during the first wave in the UK. And this is the context in which trying to actually find out whether any treatments work or not was happening. There are overstretched health services with shortage of beds and staff, concerns about whether there'd be enough ventilators, huge time pressures for staff who were busy dealing with a pivot towards the pandemic, but also very large numbers of unwell and anxious patients who would need to be approached to agree to take part in research. Just a little bit about the pathophysiology of COVID, because it really frames the different treatments that have been tried. This is just a reminder of what the disease looked like that we were seeing in 2020 and 2021. but is very much rarer now. For those that aren't familiar with looking at chest X-rays, there's a normal one on the left, and on the right you can see somebody coming in with fairly typical changes of severe COVID, with widespread patchy infiltrates. And this led to this sort of paradigm of COVID-19 pathophysiology, which pre-existed COVID and was thought to apply to other viral causes of pneumonia beforehand. And I put this up not because it was known to be true when this was actually published in May 2020, but because it's turned out to be true, at least in the sense that it sort of informs our understanding of how COVID treatments worked. But at the time this was written, it really wasn't clear at all that this was true in the sense that it would help inform the way to treat the infection. And this sort of graph shows somebody developing severe COVID with sort of time along the bottom, and then the severity of their illness sort of represented with this sort of increasing line. And the idea was really that early on in infection, this is a virus that nobody had any useful immunity to when it was first spreading. So in most people, it would replicate very actively and also could replicate in the lower respiratory tract. And then in most cases, people would develop an adaptive immune response and clear the virus. But in a fairly significant proportion of people, that would be associated with a very vigorous and disruptive host inflammatory response. And the idea that viral replication was the only important thing early on in infection, and this inflammatory response causing lung damage and then respiratory failure, and in some cases death, was important later on, has really aligned quite well with what we now know of COVID. But in early 2020, there was a huge amount of therapeutic uncertainty. There were lots and lots of different candidate drugs. Lots of people held very strong opinions about whether things worked or didn't work, but there was really no reliable data. So we had data from, after a little while, uncontrolled case series. Then later on, we had randomized trials, but lots of them were too small to be conclusive. And in this context, lots of opinions about the effect of therapy. For example, here from Trump or Bolsonaro, were actively unhelpful in terms of sort of overstating what we actually knew about treatment. And now I'm going to talk a bit about the sort of landscape of proposed treatments and categorize them a bit, just to make sense of some of the results that I'll talk about. Obviously, one of the most obvious treatments are antivirals, and they can be divided into small molecule antivirals or antibody therapies. And in small molecules, when COVID first arrived, we didn't have any drugs, obviously, that were developed for it or known to be active against it. But by screening large numbers of compounds, it was found that some things that had already been used in medicine had an antiviral activity. And so we call them repurposed antivirals. Examples being hydroxychloroquine, azithromycin, ivermectin. And just in terms of the color coding, the red ones are the ones that were tested in recovery. So you can see that the advantage of a platform trial designed for recovery was that we could test quite a wide range of treatments across quite a few different categories. There were some antiviral drugs that were already developed for other viruses, but were also found to have activity against COVID, such as remdesivir, molnupiravir. And then later on in the pandemic, there were novel antivirals that were developed specifically against SARS-CoV-2 itself. And probably the best example of that is Paxlovid. Neutralizing antibody therapy against the spike protein was also a very attractive early candidate. And early on, the only antibody therapy that was available was convalescent plasma, because as soon as people have started recovering from infection, you can take convalescent plasma from them and potentially use it as therapy. And then a bit later on, we also had specific monoclonal antibodies. So the other large group of treatments was immunomodulation, based on that idea of the pathophysiology of COVID. And most people's favourite sort of immunosuppressant is glucocorticoids, and certainly in lots of the early studies we're looking at corticosteroids and in recovery tested dexamethasone. But also there was a lot of interest in more specific immune pathway inhibitors, things such as IL-6 inhibitors, tocilizumab, Janus kinase inhibitors such as baricitinib, inhibition of the inflammasome with colchicine, and also lots of others. And lots of these medications were developed for autoimmune conditions like inflammatory arthritis, but were then turned to see whether they work on COVID. Another approach to immunomodulation was antiviral cytokines with interferons, which are part of the innate response to viruses. I'm not going to give any more details about those, although they didn't turn out to work very well. And then finally, there's a more of a sort of miscellaneous group of therapies. So it was recognised early on that the coagulation system is activated in COVID, so antithrombotic treatments have been looked at, including anticoagulants and antiplatelets. And then a very large mixed bag of other treatments, such as things affecting the host metabolism, vitamins, probiotics. And then one thing I'm not going to talk at all about anymore is non-pharmacological therapy, which is something that's had developed a lot between the first and the second waves. Things like methods of delivering oxygen, ventilation strategies, whether to prone people and that sort of thing. But it's not really my area, and it's not something that we tested in recovery. But this is just giving you an idea overall of all the treatment trials that have been registered. And you can see that there's a huge number of trials, an enormous burst of interest in COVID research, in early 2020. And actually, I mean, as of January this year, about 3,500 treatment trials were registered. And that would have included things that were set up and started and recruited patients, but just haven't, to things that were sort of registered, but maybe never even gone off the ground. But fewer than 600 of these have ever actually published results. I think this graph shows the huge number of different therapies that have been tried. You can see antiviral treatments, one of the most promising things. I mean, here is hydroxychloroquine, which there was a lot of interest in early on, but you'll see in a minute, if you don't know already, why that sort of tailed off fairly sharply in the middle of 2020. And it doesn't come out very well, but there's a wide range of things here, including anti-malarials, vitamins, all sorts of different things that were proposed. And most of these, we never got a sufficient answer for. And most of them weren't very promising to start with. But of the 500 or so trials that have actually reported results, even those, there's a big difference between. Now, I'm going to focus just now on inpatient trials, excluding trials done in people before hospital with early infection. And this shows all sort of 450 odd trials that have reported results. And each column here, from arranged in order from 1 to 450, is an individual trial result. And the number of participants in that result is plotted on the y-axis. And you can see that actually more than half of trials that were reported had fewer than 100 patients. And really, the crux of why recovery has had the impact that it has, and why, and hopefully justifying why I'm going to sort of focus talking about it, is that you can see that recovery alone as a platform trial has generated almost the top 10 results in terms of numbers of participants recruited. There's another trial, a platform trial called Solidarity by the WHO, which was also very informative. But recovery is sort of really outstanding in terms of the number of participants that we got into the trials, and that's really what's meant that it's produced clearer answers. Now, some of these trials will be deliberate sort of phase 2 trials that might have produced very clear results for surrogate outcomes. But actually, the majority of them were trying to answer the question as to whether this treatment works or not in general in hospitalised patients. And for that purpose, nearly all of these would have been completely inadequate. And also it would have been foreseeable that they were inadequate and didn't really move us forward at all in terms of our understanding of the treatment of COVID. So just a bit about recovery. I think one of the things that really was important in making recovery a success was that it was a simple, streamlined platform trial. I wasn't involved in the trial when it was being set up, but it was set up very consciously to be as simple as possible, so that it had simple eligibility, including the large majority of patients that were hospitalised with COVID, and really to try and fit into routine care without any additional demands on research staff or clinicians, and without the need for any sort of particular follow-up that's out of keeping with the kind of information that's collected normally. And that's been one of the things that's really key to its success. One of those decisions was that we were going to focus on an important outcome, and that's mainly mortality, but also the use of ventilation or the duration of time that people are in hospital. It's randomised, which, as I will mention in a minute, is absolutely crucial to producing clear answers. And then follow-up was really quite streamlined as well. And you can see this example here. There's it's an example of the consent form, which is relatively straightforward to make it simple for participants that are being bombarded with things in hospital. this is the randomization form in the middle that doesn't collect much information. It wouldn't just be routinely collected, and doesn't ask for all of the information in somebody's medical record, because we know that most of it is not going to be very helpful. And then on the right is the initial randomization scheme that they introduced in early 2020. So most patients eligible if they're in hospital, and then if they consent and their doctors agree that these treatments might be appropriate, they get randomized between these 4 potential treatments or no additional treatment. And then we collect fairly simple information about them as an outcome. And these sort of treatments listed here have evolved continually over time. Actually, now we've moved mostly to a factorial randomization system, which means that each individual treatment you can be eligible for or not, and they're all entirely separate randomizations. So if there are 3 treatments that might be reasonable to try, in the opinion of your doctor and yourself, then each one is like a separate toss of a coin, and you could be allocated anything between 0 and all 3 of them. And that's very powerful, because it means that if somebody was involved in 3 treatment comparisons, then they would contribute 3 times as much information as to whether the treatments worked or not, as if there was only involved in 1. But then moving on, another thing that helped the streamlining process and was also necessary given the conditions of the early pandemic was that it was done very much online, and the website had all of the trial documents that were required. Training was done online with sort of short videos, just to sort of communicate the essential information that people needed to know for their roles. And then randomization was also done via an online form, which took less than 10 minutes to complete. This next slide, I don't expect people to look at in detail, but this just highlights the NHS data that was also being used. And this is another aspect of something that sort of helps the trial be more lightweight and more streamlined, and wouldn't have been possible in lots of other countries. So there's an enormous amount of routine NHS data that's collected, and we can access those data sets in recovery to get at lots more details about outcomes than we would otherwise be able to get. And that means that we can reduce the amount of information that we ask people to fill in forms. It means that we can reduce the amount of monitoring we need to do, because we know that we can check lots of the things that entered on the forms, because we've got another source of information. And that's something that's really helpful, although I think this slide will probably demonstrate that although the NHS is one organisation in theory, in practice it's lots and lots of different data sets, and there's lots of division between the different nations in terms of the way things are done. So this isn't straightforward, but it's something that's been really helpful and possible in the UK to allow us to streamline the trial and make it easy for people to randomise. And then this is the sort of outcome of those design decisions that I mentioned. One of the clearly things that has been essential to the success of recovery is the fact that it's been a sort of whole NHS endeavour, and not only whole NHS, but also all of the clinical research networks and the research nurses that were working in recovery. And it was prioritised by the government and the chief medical officers, and that really helped it be taken on board within the space of a few weeks by over 150 NHS trusts. And on the left here, you can see the circles are proportionate to the amount of recruitment in each centre, and there's recruitment across the whole of the UK, and lots of these centres that are recruited, lots of patients, 1,000 patients in North and South Tees, not necessarily somewhere that would be a sort of tertiary academic centre in lots of other senses, but it's almost every busy hospital was recruiting extremely well in recovery. And on the right, you can see the daily recruitment. Along the top, these bars represent which treatments were being tested in recovery at each time, and that platform trial design means that as treatments were dropped out, new ones came in, and the fact that there was lots of recruitment meant that we could essentially drop treatments when we found a clear answer. So there wasn't a set recruitment target. The data monitoring committee could look at sort of blinded data and then see, is there clear evidence that this works, or is there clear evidence that it doesn't work? And also that there's the statistical uncertainty is enough to say that not only did we not find a positive result, but it's inconsistent with this really being helpful. And that's the thing that's really made recovery results much clearer than most others. And this recruitment graph in recovery mirrors the epidemiology of COVID in the UK, with a big first wave and then the second wave. And at points during the second wave, we were recruiting over 500 people a day into the trial. And and I'm aware that for some of these comparisons, I mean, hopefully some people on the call have been involved in one way or another in recovery, because it's involved, all sorts of clinicians, but also people in pathology. So much for recovery in terms of design. I'm just going to talk now about a few specific treatment questions and the results, and how sort of the randomized trial evidence fits into us sort of answering them. And hydroxychloroquine's got quite a bad name because it was advocated quite strongly by people without very good evidence. But it's worth going back to 2020 and just remembering that actually it was a promising treatment. It's one of those repurposed drugs that I mentioned where in vitro data suggested that it had a useful antiviral activity, although at the upper bounds of the concentration at which it was effective was quite high compared to the doses that people would normally have taken. This on the left just shows a paper demonstrating that it has antiviral activity at sort of high doses. And then the other thing that you get relatively early on is observational data. So this is data looking at patients that either received a treatment or didn't as part of their routine care, so not as part of a randomized trial. And this is a report from China sort of that came out fairly early, and you can see here the survival curve of people. This is going from all surviving downwards, which is the opposite way to most of the rest of the survival curves I'm going to show you from recovery. But the people who were critically ill who didn't receive hydroxychloroquine, the mortality was about 48%. In those that received hydroxychloroquine, it was about 19%. And their conclusion was all 550 patients received comparable basic treatments, including antiviral drugs and antibiotics, and about 50 of them were treated with oral hydroxychloroquine, and the fatalities in the hydroxychloroquine group were significantly lower than those in the no hydroxychloroquine group. And this is the kind of evidence that's been generated lots and lots during the COVID pandemic, and it's often very counterintuitive. So you can probably see where I'm going with this, because we all know that hydroxychloroquine turned out not to be an effective treatment. But if you look at this paper, the characteristics of the patients in both groups appear identical, and yet we know from previous experience that observational data can be surprisingly misleading, and it can produce results like this because of biases in that the two groups weren't really comparable to start with, but they're often not comparable in ways that you really don't know or can't adjust for. But if you imagine yourself as a clinician or a patient, or the family of a patient who is critically unwell, given this information for a drug that there's lots of evidence for and relatively non-toxic, taken by hundreds of millions of people, it actually takes quite a bit of bravery to still know that randomization is necessary to really answer the question clearly. But luckily in the UK, we've got a good tradition of evidence-based medicine and thorough research, and so the first really reliable evidence on whether or not hydroxychloroquine works came from recovery, and this shows the effect of hydroxychloroquine on mortality. As I mentioned, it's the other way around, so mortality starts at 0 and then goes upwards over 28 days. And those patients that were allocated hydroxychloroquine are in red, and those allocated usual care alone are in black. And with about 5,000 patients randomized, it was clear that hydroxychloroquine really added nothing in terms of reducing the risk of death. In fact, if anything, it was non-significantly adverse. And when you get really clear results like this, it changes practice very quickly in a way that smaller trials or observational studies just can't do. So these results were announced on the 5th of June, which was trial day 78. And then within 10 days, the FDA had revoked a previous authorization for the use of hydroxychloroquine. And just to prove that this isn't just an effective recovery, Solidarity also was studying hydroxychloroquine. This is the WHO trial that had results for hydroxychloroquine in about 2,000 patients. And again, when you do a proper unbiased experiment with reasonable numbers, you get the same answer. And they also showed that hydroxychloroquine was not beneficial and might actually be harmful. So then, soon afterwards, on a more positive note, there was the first result from any trial in the world that really showed a treatment that clearly saved lives in people hospitalised with COVID, and that's corticosteroids. So in recovery, 6,500 odd people were randomised to dexamethasone or usual care alone. And on the sort of panel on the left, you can see the effect on mortality, and it looks pretty modest on this because it goes from a scale of 0 to 100% mortality. But this is statistically a very clear result. So there's no question that this could have been due to chance. And actually the difference between usual care and dexamethasone, on death from COVID was really worthwhile. But the thing that was slightly unanticipated was that the result depended very much on the degree of lung disease. And it became clear, looking at the result, that there was no benefit in people not requiring oxygen. If people needed oxygen, then it reduced mortality by about a fifth. But in those sickest patients with the worst lung disease, it reduced mortality by about a third. And I'm just going to show this here in different panels. It's a bit of a messy graph, but you can see in the top right are people on invasive mechanical ventilation. And in those, it reduced the risk of dying by about, as I said, a third. And because the risk of death was so high, the number of people needed to treat to save a life was only about 8. And this is for a treatment that cost a few pounds. And then, conversely, on oxygen, there was still a very clear benefit, very worthwhile having. In people that hadn't developed significant lung disease, it really didn't have much of an effect, could even have been harmful. And this actually is, is in keeping with that sort of paradigm of pathophysiology that I mentioned earlier, where the inflammation is causing more and more problems with people with severe lung disease. So these results were announced in June. So again, within 3 months of the trial starting. And not only did they offer a treatment that was worthwhile for COVID, it was particularly nice because it's a cheap and globally available treatment that's in every hospital around the world. And also, it sort of proved the paradigm that immune-mediated lung damage is a modifiable drug target. Because before this, lots of people, including me, were very concerned that steroids might actually worsen outcomes in COVID because giving an immunosuppressant when somebody's got an infection is a sort of slightly worrying thing to do. And there were people that wrote to the trial investigators saying that it was unethical to do this trial because it clearly would be damaging. But by randomizing, you produce clear results, and with large numbers, it can change practice. And actually, NHS guidance changed the same day to recommend using steroids in people hospitalised with COVID. Given that immunomodulation was effective, the next question is whether there's any other things that could be done to improve the sort of effective immunomodulation and reduce the risk of death. And another immunomodulator was tocilizumab, this is a monoclonal targeting the IL-6 pathway. And Recovery randomized 4,000 patients requiring oxygen to this comparison, and it showed that it reduced the risk of death by an additional 15%. So by this time, essentially everybody in this group was receiving steroids because that had become standard of care. But actually tocilizumab reduced the risk, again, highly significantly. And just to sort of explain, I guess, why Recovery has helped to move things forward in a way that lots of other trials were unable to, I'm just going to show you a summary of the different trials that existed at the time of publication when Recovery published these results. So this is a forest plot of trials, and in case you're not familiar with looking at these, each line is a different randomized trial. And you can see the number of people involved in each one, and you don't need to look at this in detail, but essentially the size of each square is proportional to the number of people involved in that trial. And the uncertainty around the estimate as to whether it works or not is represented by the lines. And so then if you add together all of the trials that have been done up until the point that Recovery published this result, you can see that there was some trials, well, one trial, REMAP-CAP, had a positive finding for tocilizumab already before Recovery was published. But other trials had found negative, borderline significant results against tocilizumab. And the overall, adding all of the worldwide evidence together, was that it looked promising, but it was consistent with either a real reduction in death that would have been very worthwhile having or no effect. And then Recovery added much more information than all the other previous trials combined, and really showed that actually this was a life-saving treatment. And again, by producing really clear evidence, it immediately moved things forward. Tocilizumab could be used in routine practice. The question was answered, and people could move on to other questions about the treatment of COVID. And finally, in immunomodulators, there's another drug called baricitinib, which targets a different pathway involving Janus kinase. And this was the sort of last positive result from Recovery in terms of immunomodulation. And in this, we randomized 8,000 people requiring oxygen. And this showed that there was an additional reduction in risk of death by about 13%. And this was almost all of these patients were receiving corticosteroids, and quite a large proportion were receiving tocilizumab, but it seemed to be the same regardless of that background treatment. And once you start sort of multiplying each of these reductions, it turns from sort of fairly moderate effects into quite a large reduction in the risk of death in aggregate. So probably now, for the sickest patients, we could reduce their risk of death by half if they have kind of modern immunomodulator therapy. And again, so like with tocilizumab, there have been several smaller trials beforehand. In this case, actually, they'd shown that there was a clear reduction in mortality with baricitinib. But all of them individually were too small to really give very much certainty as to how much that was. And recovery really just produced confirmatory evidence that really demonstrated that this was another effective treatment. So now I'm just going to move on to antiviral treatment. Oh no, actually no, sorry, there's one last thing to say about immunomodulation, which is that the question obviously arises as to whether there's any limit to how much immunomodulation could benefit people in terms of treatment. And not to the world can't afford baricitinib or tocilizumab. Most of the world can afford corticosteroids. So after demonstrating the effect of 6 milligrams of dexamethasone, we went on to look at whether higher doses of dexamethasone might be beneficial because they have bigger effects. And this actually showed that higher doses of dexamethasone could be harmful in patients that were only requiring simple oxygen. There was a clear excess of mortality if using higher doses. So we randomized 20 milligrams versus 6, and the comparison was actually stopped by the data monitoring committee because it looked like it was harming these people. In people that have the highest level of ventilation sort of support, the question is still open. So it didn't appear to be as harmful in them. But this is just another sort of demonstration that you need randomized evidence to really answer these questions clearly. So moving on to antivirals, convalescent plasma was a very promising treatment that's been used for over 100 years. And I mention this because it's also quite an instructive trial result, and because it was a trial result that relied on transfusion laboratories up and down the country who were instrumental in distributing the convalescent plasma and in handling the samples that allowed us to produce a clear result for some of these therapies. So the rationale is that as antibodies help clear infection, for people who have recovered, they've got very high anti-COVID antibody levels, and these can be transferred into people who are at risk of dying from disease. It may be true that people are at highest risk if they hadn't yet developed their own antibodies. So we measured antibody levels at baseline in people who were receiving convalescent plasma. And in the UK, it was never really established in treatment outside of randomized trials. And in contrast, in the US, they also had a lot of interest in convalescent plasma. But they sort of went ahead and started a convalescent plasma program without any randomized evidence of benefit. And in the end, treated about 500,000 people that were hospitalized with COVID with convalescent plasma. And they thought that it wasn't going to be possible to do a randomized trial, and that they would hopefully just get this treatment out to people because it might work, although it's quite expensive and difficult to produce the plasma. And they thought that hopefully they could find out with observational studies whether or not it was actually helping people. So this is looking at people that either received the treatment or not, and then find out whether or not they died or lived. And hopefully that way would find out whether or not the treatment was effective. And again, you can probably guess where I'm going with this. This is some results from the US fairly early on that were very influential, supporting the use of convalescent plasma based on an observational study, not a randomized study. And they actually had quite a nice design, which is that the people giving the plasma didn't know how much antibody was in it. That was measured and determined afterwards. And so by comparing people that received what turned out to be low-dose convalescent plasma versus high-dose convalescent plasma, you might be able to tell whether or not the convalescent plasma was having an effect. And they did this analysis and very confidently said that it reduced the risk of dying by about a third, and it was highly statistically significant. Luckily in the UK, we've got a tradition of evidence-based medicine, and we're in a position to randomise because of the efforts of NHSBT and all the people collaborating in the trial. And this really found that there was no appreciable effect of convalescent plasma on the risk of dying from COVID in people admitted to hospital. So this is, the risk was 1.00, and it really ruled out any meaningful benefit from this treatment. We randomised 11,000 participants and nearly 3,000 of whom died. So the risk of death was very high, and it didn't seem to work. It didn't seem to work in any particular group of people, and really didn't convincingly work even in those people who had no antibodies at baseline. And again, this is the last forest plot that I'll show you, but just to sort of re-emphasize this summary of all the trials that existed on convalescent plasma before the recovery result looks quite similar to that of tocilizumab. About 10 trials, some of them had, one of them had nearly 500 patients in, including 1,600 people in total. So, a very large exercise to try and, get the randomized evidence that was here, but it was completely inadequate to answer the question as to whether convalescent plasma worked at all or not. So, one trial that had a significant effect in favour, and, in aggregate, they suggested that it might reduce the risk of mortality, but it was consistent with convalescent plasma reducing the risk of death by 40%. So, almost a miracle treatment in a way, to having no effect at all. And it's just, that the random error is so unforgiving that even with trials of this size, we just weren't able to tell whether or not convalescent plasma was actually helpful. And then when you add in recovery with a completely null result, in aggregate, most of the information was from recovery, and it showed that convalescent plasma really had no effect. And I think in recovery we were using high titer convalescent plasma. We were giving people 2 units. It was a really, really good test of exactly the kind of treatment that ended up being given to 500,000 people in the US, and it really didn't help. And even people that were very keen on exploring convalescent plasma after this trial result, they started focusing on whether given earlier it might be helpful, or with very, very high doses it might be helpful. But by producing clear evidence, just suddenly everything moves on. So now I'm just going to talk about monoclonal antibodies. This is the one slide where I'm going to mention outpatient treatment, which is another huge success of COVID therapeutic research. And monoclonal antibodies developed against the spike protein are capable of delivering much higher doses of neutralizing antibody than convalescent plasma was. And when these were developed, I think in late 2020, it was discovered that if given very early in infection, they could reduce the risk of hospitalization and death by a lot, by about 75%. The problem is, in order to get that benefit, you need to treat huge numbers of people, which is not the case in hospital. But it was clear that, again, in keeping with that sort of paradigm of pathology, early antiviral treatment is very effective. And subsequently, similar effects were also seen with small molecules, so Paxlovid and remdesivir. And this survival curve is just an example of one monoclonal antibody, which shows that it prevents people from being admitted to hospital, or is very effective at doing so. So then the question arises, whether this is helpful in people who are hospitalized, who are at very high risk of death. And this was another comparison that we looked at in recovery. So it was unclear if hospitalized patients would benefit from monoclonal antibodies. Again, we thought that people that didn't have their own antibodies yet, when they were admitted to hospital, might be most likely to benefit. And in this case, that was a pre-specified analysis. And we found that actually in seronegative patients, this monoclonal antibody cocktail called Ronapreve reduced the risk of death by about a fifth, but had no effect in people that had their own antibodies when they were admitted to hospital. And so here you can just see a slightly more complicated graph. Up and top, the 2 lines are those who are antibody negative. You can see being antibody negative also means that your risk of death is very much higher. And with treatment, we reduce that very definitely. These 2 lines are those people that were seropositive when they came into hospital. And you can see that there was no effect in them, which is good. And we started using them for a while in the UK. Again, as soon as these trial results were released, quite soon afterwards, this became available as a treatment on the NHS. But the problem with monoclonals is that spike mutations can lead to dramatic loss of activity. And actually, not very long after this started being used, the Omicron variant turned up, which on the plus side seemed to be less virulent, so it was causing less severe disease. But unfortunately, it was completely resistant to this antibody cocktail. So this has fallen out of use. That's all I'm going to say about individual results. Just to sort of summarize, by having an efficient design with factorial randomization, by recruiting large numbers of patients, recovery's managed to produce much clearer results in terms of COVID treatment of people in hospital than any other trials. This is a list of all the sort of hits and misses from the trial. There's a fairly good hit rate. Most of these results I didn't show you, but the number here for each therapy is the number of people that are involved in that comparison. And I'll just go back to sort of mention that factorial design means that although the trial's done extremely well by recruiting nearly 50,000 people, we've actually, each person has contributed on average to two questions. So effectively, it's equivalent to trials adding up to 93,000 people. So in terms of recovery now, there is still, some people dying of COVID. It's still an important question, but just relatively much less important than it was a couple of years ago. But recovery is still looking at the treatment of COVID, and hopefully it's gone away as a major public health problem, but we can't be completely certain. So we're still looking at that. During this period, recovery has expanded to other countries, and we're now active in 7 non-UK countries. And we'd like to try and keep this platform going to look at other infections, including now we've just started looking at treatments for influenza like Tamiflu and steroids. I mentioned at the start that our understanding of COVID has leapfrogged most other conditions, and we don't even know whether steroids work in flu, which is kills lots of people and has been around for a long time, or even bacterial pneumonia, which kills 2 million people a year, and has done so since time immemorial. But we've never had similar trials that could actually answer the question for other common conditions. So it'd be very nice to start addressing some of those. And then final point is just to kind of bring back some of those points about trial design. And the thing that's really distinguished recovery from most other trials, and I'm not suggesting that all useful results have come from recovery, but the thing that's really made it different is that it was designed, and because of the circumstances of the pandemic in the UK, was able to identify or exclude relatively moderate effects on the risk of death. And by moderate, I mean a reduction, for example, of a 1/6 or a 1/5. So not really very moderate if you're the person involved, but beyond the reach of most trials that recruited only hundreds or a few thousand patients. And in order to work out whether or not treatments were having those effects, you need to avoid moderate biases, and that's why randomization is absolutely necessary, because it's almost impossible to get rid of the biases that are associated with observational studies. But you also need to reduce random errors, which is why you need large numbers. And then beyond that, there are only a few relatively simple things that are necessary. So you need to make sure that people do have good adherence to the treatment, you get good follow-up, you analyse by allocated treatment, which I won't go into, and then always you should think about trials in the context of all the other trials that have been done on that question. So you need to look at meta-analyses rather than just single trial results. And that's all I was going to say. So thank you. Sorry, I've gone slightly longer than I meant to, but I think there's hopefully a bit of time for questions if anybody has.
No problem. Thank you very much for that. Very, very, very interesting indeed. I think we've got one question. I think, and as Andrew is one of our speakers, I think he's got the privilege to speak now. Actually, so Andrew, do you want to ask your question? We can hear you, Andrew.
Can you see me?
We can't see you, but Dory, ask your question.
All right. Thanks, Leon. Fantastic talk. A couple of questions. Firstly, we know that erythromycin doesn't work, but what was the rationale way back when for people attempting to try an antibiotic against a viral disease?
I think it was just the same as the other repurposed antivirals that showed an in vitro effect, and I think that had been noted with some of the things. But again, it's I mean, often people talk with azithromycin as the main one that people were thinking about. Sometimes people talk about anti-inflammatory effect, but this was mainly just because it had an in vitro antiviral effect.
Okay, thanks. I'm speaking later on vaccines and viruses, and clearly in my talk I'm going to be talking about the monoclonal antibodies you just described. I'd like to hear your views and therefore preempt some of the questions to my talk about whether the monoclonal antibodies raised against the Wuhan strain are still valuable in terms of all the variants we've had since.
It's a very good question, and there's a lot that could be said about the effect. I mean, it's been incredibly instructive, this pandemic, watching the succession of variants, which has been spectacular in itself, but then also looking at the effect those have had on monoclonal antibodies. And I think nobody anticipated at the beginning the extent to which successive variants would wipe out antibodies. And this is mostly not sort of selection pressure by the antibodies. It's just that when they change, you can just lose antibodies at a stroke. I mean, so some of the antibodies, one of the antibodies that we're still testing in recovery was raised against the initial strain, sotrovimab, and it's still got some useful activity, but that's less than it was previously. Most of the others have been wiped out, so it's a real problem. You know, they're an incredibly promising therapy, but I don't know what the solution is to this, you know, this sort of fact that resistance, you know, can develop so quickly. I mean, maybe cocktails ought to have been tried initially. You know, if there was another pandemic, maybe we ought to be trying to look at cocktails of 5 or 6 antibodies, because they've just, you know, it's just made them impractical to use, and impractical, I guess, for companies to develop them. The fact that they disappear so quickly.
Yes, I like the idea of a cocktail. That's against different parts of the RBD, I guess.
So I mean, well, I mean, actually, the RBD mutates very quickly, and so most of the antibodies that have been developed against the, you know, the actual receptor binding motif have been wiped out. So sotrovimab binds to a different part of the RBD that isn't involved in sort of non-covalent interactions, and so might be a reason that it lasted a bit longer. But yeah, they're just, I mean, the evolution is just very, very brutal for individual monoclonal antibodies.
And finally, have you got a really famous father, uncle, or grandfather?
Yes. Well, relatively, but it's in terms of
lucky you, I hope you inherited some of their genes. Right. Thank you very much for your contribution, Leon. It's been great. Thanks, David.
Thank you, Neil, and thank you for those questions, Andrew.